One of the most intriguing aspects of recent NAD+ research is the suggestion that restoring NAD+ levels might not just slow decline but actually support functional recovery in damaged brains. This is a significant departure from the previous assumption that neurodegeneration is a one-way street. Animal studies have shown that when you boost NAD+ in mice with Alzheimer's-like pathology, you don't just stop further damage. You actually see improvements in cognitive function. The brain starts working better even though the plaques and tangles are still there. The mechanisms behind this potential recovery involve several NAD+-dependent processes. First, DNA repair. Neurons are constantly accumulating DNA damage, and while they can't divide to replace themselves like other cells, they can repair damage if they have the resources. NAD+ is essential for multiple DNA repair pathways. Second, synaptic maintenance. Synapses, the connections between neurons, require enormous metabolic support to maintain. NAD+ helps fuel the protein synthesis, vesicle recycling, and structural remodeling that keep synapses functional. Third, mitochondrial quality control. Cells use NAD+-dependent processes to identify and recycle damaged mitochondria while making new healthy ones. So could diet support these repair processes? In theory, yes. If you can maintain healthier NAD+ levels through better nutrition, you might be providing neurons with more resources to invest in repair and maintenance rather than just barely surviving. But we need to be extraordinarily careful about overstating what this means in practical terms. The animal studies showing cognitive recovery with NAD+ restoration typically involve giving mice NAD+ precursors at doses far higher than you could possibly achieve by eating niacin-rich foods. They're also giving these compounds to relatively young mice with early or moderate pathology, not to very old mice with advanced disease. And mice aren't humans. Their lifespans are measured in months, not decades. The cumulative damage in an 80-year-old human brain with Alzheimer's is vastly greater than anything you can model in a mouse. There's also the question of whether raising NAD+ through diet can even significantly increase brain NAD+ levels in humans. NAD+ itself doesn't cross the blood-brain barrier. The brain has to make its own NAD+ from precursors. Those precursors can cross into the brain, but the efficiency of this process and whether dietary amounts are sufficient to meaningfully raise brain NAD+ remains uncertain. We have some evidence from supplement studies using nicotinamide riboside or nicotinamide mononucleotide, but these are at doses much higher than food provides, and even then, the evidence for cognitive benefits in humans is limited. The concept of nutritional support for brain repair is appealing, but it runs headlong into the reality of how extensive the damage is in Alzheimer's disease. We're not talking about a few injured neurons that need support to heal. We're talking about widespread neuronal death, massive synaptic loss, profound metabolic dysfunction, and chronic neuroinflammation. The idea that providing better nutritional support for NAD+ production could allow meaningful functional recovery in this context is, frankly, a long shot. Where this approach might have more potential is in earlier stages of cognitive decline, mild cognitive impairment, or very early Alzheimer's, where there's still substantial neuronal mass that's dysfunctional but not dead. If those neurons are energy-starved but structurally intact, then improving their energy metabolism through NAD+ support might help them function better. This is speculative, but it's at least biologically plausible. There's also the possibility that for cognitively healthy older adults who have age-related NAD+ decline but no disease, dietary strategies to support NAD+ might help maintain cognitive resilience and slow the trajectory toward decline. This is prevention blurring into early intervention, and it's an area where dietary approaches might actually have their greatest impact. The bottom line is this: NAD+ is essential for many repair processes in neurons, and better nutritional support might help maintain these processes. But the gap between might help maintain repair capacity and can reverse established brain damage is enormous. We need much more human research before we can make strong claims about diet's ability to support cognitive recovery through NAD+-dependent repair mechanisms. Until then, this remains an interesting hypothesis with biological plausibility but limited clinical evidence.